This year, we actually had the fourth World Parkinson's Congress, and Dr. Stossel was primarily involved in the organization. There were a number of hot topics that were presented there. These included things like inflammation in Parkinson's disease, the microbiome in Parkinson's disease, use of wearable sensors to track the disease, the genetic architecture or the genetic influences in Parkinson's disease, and some talk about stem cells. Now, I'm very pleased that rather than the stem cells, we are involved and interested in all of these. So it just tells us that our intuition of where are the proper directions that we should be heading, I think are largely correct. One of the things that came up in the past year that was quite exciting is the potential for a blood test for Parkinson's disease. This is from a group out of Melbourne, Australia, where they looked at a particular protein from a certain type of blood cell called a lymphoblast. And they were able to detect that in people with Parkinson's disease, the mitochondria, which are the energy stations in the cell, these are ramped up significantly in people with Parkinson's, and this can be detected with a blood test. What's required, though, is to see how specific this is for Parkinson's, or if it's just a more general manifestation of other types of neurodegenerative disease. But certainly that would be an exciting advance. Another interesting study that came out in Taiwan, where they had a very comprehensive registry of people who had various types of infections, and particularly hepatitis. It almost seems like the GI is very important for the brain health. And what they found was that people who had hepatitis C went on to develop Parkinson's disease at a significantly greater rate, but not the other types of hepatitis. Again, this gets into the complex etiology of Parkinson's, which is probably, again, a complex interaction of genetic predisposition and environmental influences. And some of the environmental influences might include infectious processes. We all know that the alpha-synuclein is the protein that clumps in brain cells in Parkinson's, and so there's a lot of interest to see whether or not this abnormal protein could be detected in easily accessible tissues such as saliva and so on. So our colleagues in Toronto, Dr. David Munoz, is looking at alpha-synuclein from biopsying the salivary glands, which are just available under the chin, from the colon, or from the skin. There's a Michael J. Fox Foundation study that's called the S4 study, and in addition to these other areas, they will be looking at saliva, blood, and spinal fluid. And this might be, again, a biomarker for both diagnosis and tracking the progression of the disease. The other thing which I think I just mentioned, this has been going on for a couple of years, but I think it's an excellent model of the way things are going. It is becoming apparent that if we want to look at some of diseases like Parkinson's, we can't just look at 10 or 20 subjects. We have to look at thousands, and that's certainly the case with genetics, but most of our studies that involve imaging typically involve dozens of people because imaging is quite expensive. So what the Michael J. Fox Foundation has done is they've collaborated with a number of private partners and set up the PPMI, which is the Parkinson's Progressive Markers Initiative. And what this is that centers from around the world have data, which might be imaging data, and they upload it to a central database. And then people from around the world can look at this pooled data. And having these great large number of samples makes some analyses possible that wouldn't be otherwise possible. Now, Dr. Cresswell and I contributed to this in a small way, but what we did is we took these hundreds of samples of people with brain images from people with Parkinson's around the world, and we tried to look at an old question, and that is, we know that Parkinson's tends to start on one side of the body. And what wasn't clear is if you're right-handed and it starts on the right side of your body, is it worse or better than if it starts on the left side of your body and vice versa? And because the number of people who are left-handed is relatively small, it's very difficult to answer that question if you only have 20 subjects. But if we have 300 subjects, then we can start to answer these questions. So with our colleagues in SFU, Simon Fraser University, we're able to look at changes in cortical thickness and answer these types of questions. So it's not this particular study that's important. I think it's this concept of pooling data across centers and then having the data available for researchers from around the world to download and provide novel analyses. I think that's an excellent model and something that we will see more of in the future. There are a number of advances in the fundamental mechanisms. So this is a paper from Dr. Farrer's group, so I won't spend a lot of time because he will be speaking later. But one of the biggest challenges, of course, in Parkinson's is the variability in the rate of progression. And even if someone has a known genetic mutation, the onset of the disease varies tremendously. So here's a discovery where it appears that there may be genetic influences that dictate when the disease's onset. And again, I will defer to Dr. Farrer about that. So, there are a number of treatments that are in different phases of studies. So just as a review, typically when a new medication comes out, there are different clinical trials that are run at different phases. I just summarize them here so it's clear when I allude to this later. But basically a phase two study is done in a small group of people to see if there's any effect of the drug. Assuming there is an effect, then they go on to a phase 3 study in a large group of people, and that then determines, what's the optimum dose range, what's the size of the therapeutic effect, and so on. So one of the things that is interesting is the new way to deliver medications that have been around for a long time. So there is a new delivery method that's been available in Europe actually for several years, but is just becoming available in Canada. It's called Duodopa. It is a intestinal gel. It's levodopa preparation that's in a gel form, and it's continuously infused via a pump. And this is for people who tend to fluctuate throughout the day between wearing off and having excessive dyskinesias or writhing movements. And these so-called brittle people who find it very difficult to get the exact right level of medication, this can be handled by inserting a small tube through the abdomen and then having a pump that continuously infuses this gel. We've been having some challenges of having it being covered by the BC government. It is available in several other provinces. And I see Jean Blake from the Parkinson's Society of BC in the audience, and we are currently trying to advocate coverage for a select subset of people. There is a company called Neuroderm, which is interested in a drug called apomorphine, which is available in the US, but not available yet in Canada. But this is a medicine that's normally given by subcutaneous injection. And what they have developed is a patch, which is basically several little micro needles that would directly infuse this medication. So this is another delivery method of an old drug. And finally, there is a Canadian company that's doing a phase 2 trial on a sublingual film. Many of you may be familiar with those Listerine films that you can put on your tongue. Well, this would be a way to deliver apomorphine medication under the tongue. The other area that's come up, which has been quite interesting, is retooling medications that are already out there for Parkinson's disease. One of them is nilotinib, which is, many of you heard on the news, was an anti-cancer drug, which was shown to be effective in Parkinson's. Unfortunately, there was so much hype about this medication that it was sometimes hard to isolate the truth from the hype. It does look like they will probably have a phase 2 trial this year. The concern about this medication is it's a chemotherapeutic agent, so it has a lot of side effects, and everything in medicine is a risk-benefit trade-off, so you want to be sure that the medicine is safe, and so they will be readdressing this with a phase 2 trial, likely funded through the Michael J. Fox Foundation. There are a couple of phase 3 trials of medications that are interesting. One is isradipine, which is a medication that's used for hypertension. And what happened is when they did large epidemiological studies, they found that people who were taking this medicine for hypertension actually had a lower incidence of Parkinson's disease. Another medication is inosine, and this is a result of the observation that people who have the antioxidant urate in their blood at higher levels are less likely to develop Parkinson's. So inosine gets converted in the body to year 8. And so this is currently a phase 3 level trial looking at this. There are a number of new medications coming down targeting non-motor symptoms. So pimavanserin is a medication which is approved by the US FDA. It's not yet available in Canada, but we will be looking forward to having this medication available to us for the treatment of psychosis and Parkinson's. Buspirone is a novel medication for anxiety, and so this might be very useful for anxiety and Parkinson's. There's a new medication for constipation, which is a chronic problem in people with Parkinson's. And Reset Therapeutics is a company that's looking at new compounds that may reset the sleep-wake cycle. And this may be important for both daytime sleepiness and problems getting difficulty at sleep at night, problems that are quite common in Parkinson's. Now, what about some innovative treatments on the horizon? There's a biotech, Voyager Therapeutics, is doing a study on gene therapy, where they inject a gene and have the attempt is to try and get the brain to convert levodopa to dopamine. MRI-focused ultrasound is a technique that's used for essential tremor, not Parkinson's disease. It's currently available in Toronto and will soon be available in Calgary. But now in the States, they're also looking at this for the treatment of dyskinesias in Parkinson's, and this is being done in the US. A monoclonal antibody, so this is an infusion of a medication. And what they've shown is that if you infuse this medication, there is a reduction in the alpha-synuclein levels after a single infusion. And then looking at vaccines. So what they showed in the first clinical trial with a vaccine is that if you give like a flu shot, about half the people develop antibodies to alpha-synuclein. Unfortunately, the antibodies regressed after a year, but they were able to show that if you give you booster shots, much like you would get tetanus, that you get reemergence of the antibodies, so that's useful. And finally, there's been some interest in new healthcare models. And so this is out of our colleagues from the Netherlands. And what they are doing is they're trying to comprehensively monitor people with molecular analysis, advanced brain imaging, wearable devices with sensors, and then doing big data analysis. And I bring this up because this is very much along with what we have proposed to do as well. So what's new, particularly at the PPRC? Well, it's been an exciting year. The good news is that we've hired a couple of new neurologists, Dr. Jason Valerio and Jonathan Squires. These are excellent neurologists who have a keen interest in looking after people with Parkinson's disease. And in the case of Dr. Valerio, also looking at people with sleep problems. We're delighted to have Dr. Deborah Hawking, who's a family physician who is running a clinic as well. And VCH has also, Vancouver Coastal Health, has also increased the number of nurses that are available in the clinic, and this has been greatly appreciated and useful for the running of the clinic. As a result of this, we've been able to increase our throughput in the clinic by about 45%, which I think is quite significant. And we anticipate that this will result in significantly reduced wait times. Nevertheless, we've had some challenges. We just had a retirement of our senior nurse, Sharon Yardley. I think she's in the audience somewhere. Sharon, oh, there she is in the back. Sharon has been with the center for literally decades and probably knows more about Parkinson's than most neurologists. So it's a big loss for her retirement. And we've had a transient loss of the physiotherapist, and we're currently investigating how to follow up with recruiting a new physiotherapist. Now, I think at this point, it's useful to think about the overall vision for our center. Well, our main focus has been and will continue to be excellent clinical care. We will always try to provide the best care for people with Parkinson's, but in order to support that vision, we need access to research, and research enables us to make sure that we are providing cutting-edge care. And when you think about research, I think it's useful to think about having a balanced portfolio of research that's useful in the short term, the middle, and the long term. And the long term, we're ultimately looking for a cure, and of course we anticipate that the path to a cure may be very circuitous and may have various bumps along the road. But if we look at research that's short-term, medium-term, and long-term, short-term research will involve things such as novel treatments that we can apply to people, like exercise, for example, analyzing data, for example. Can we look at our database and see that there's gender differences in depression? That might dictate how we treat depression differently in the clinic, and also clinical care models about how we actually deliver health care. In the medium term, we've had in the past, and will continue to have, a strong emphasis on imaging. So imaging allows us to be a biomarker and assess the effects of intervention. It allows us to prognosticate, to predict how people will do in the future. And it may also stratify people into suggesting what types of therapy will be good for what subjects. And then over the longer term, we'll be looking at things like genetics and molecular biology. So I think we need to have a kind of a balance over all of these. So you will get a feel for all of the successes of the research by looking at the posters later on. But I would just like to touch on a couple that I think are particularly noteworthy. So one of the things that we've done in the past year is we've incorporated a Parkinson's disease template. And this has enabled us to capture data in a semi-quantitative way that can be uploaded to a clinical database, and it will integrate well with the Center for Brain Health that we'll hear more with from Dr. Stossel. And it also allows us to do data mining and so on. For example, this gives you a feel for this. We were able to take the data that we've already collected and then see if there were subtypes of cognitive profiles in Parkinson's. So that's something that we could do that has an immediate impact in the clinic. The other thing we've done is we've incorporated electronic aids in the clinic, such as using iPads. And this is a much more efficient than having paper-based things that have to be scanned in. We've also developed iPad apps in collaboration with Conquer Mobile, a Surrey-based company. And this is, we are currently doing this on an ongoing basis to screen cognition and motor performance. This has resulted in a bit of coverage through the media picked up by CBC, for example. One of the things that I think is very important is being the effect of exercise. So we will hear more from the effects of exercise on PEP from Dr. Stossel. But Sunny Tan in my lab has been looking at the effect of a walking program where people listen to music. And what she's shown is that there's been changes in the white matter of the brain, or the myelin, and changes in the functional connectivity in the brain after people exercise. And in fact, there's differences between whether or not you're exercising with music and without music. The other thing that we're particularly excited about is she's shown connectivity in a part of the brain called the PPN. And this is a brainstem structure which is vital for balance. And we know that balance is a big problem in Parkinson's disease that does not respond well to medication. So being able to show that an exercise intervention modifies the connectivity to this region, I think is a particularly important advance. She will have a poster on this that you can see later. Matt Ciccelli in Dr. Stossel's lab has shown some changes in PET. And I won't spend a lot of time on that because I think Dr. Stossel is going to talk about that in the next talk. Another thing is that Sujin Lee and Jaguar Kai have, with Dr. Wang's lab, have also shown that if we electrically stimulate the vestibular system, we can modulate connectivity to this PPN structure, which is associated with balance. And in fact, where we saw the changes in connectivity is exactly where prior PET studies had shown to be important for falling. So those are things that will have immediate effect, like the effects of exercise. Those are something that we can implement immediately. What about the medium term, looking at advances in imaging? Well, again, I'll defer to Dr. Stossel and Sosi, who will be telling us about tau imaging and inflammation and PET tracer spatial distributions, things that are important. These will also be part of the poster things, but I won't steal their thunder. I'll defer to that. But we have had some advances in the MRI method. So One of the things is it's always thought that the white matter, which is the wires that connect the different parts of the cortex together, was thought to be unaffected. So Robert Baumeister in my lab has shown, using a technique developed by Professor McKay here at UBC, has shown that there are changes in the white matter and that they actually predict disease severity to quite an accurate degree. So I think this might be a very exciting thing that we can look at further. Again, over the long term with genetics and molecular biology, I will defer to Dr. Farrer. For example, they have a new gene that's detected in coding an alpha-synuclein chaperone, and again, I will defer to him and his talk about that. So you can see that it's been an exciting year for the PPRC. We've had expansion of our clinical capacity. We've changed how we collect clinical data and capture it in a semi-quantitative way. We've had short-term successes where we've looked at novel treatments, the effects of exercise. We've had advances in PET imaging. We've had advances in MRI. And then we'll also hear some more about the advances in the genetics and molecular biology. Another big success story is we were successful for a CFI funding for a new 3T scanner, which we anticipate will come online at the end of 2017. And we will also hear from Dr. Saucy about the PET MRI machine, which is currently under review. So at that point, I think I will step down and ask Dr. Stossel to take over.